Welcome back to the class. So, in our chapter Strategies for Enhancement in Food Production, we learnt about some of the management techniques of the farms, both in case of poultry as well as in the cattle or the dairy farms. Today, we will talk about one more topic which is called as the animal breeding. This is because breeding of animals is a very important aspect in animal husbandry. Now, why is it important? It is because it is this technique that increases the yield of animals, and it also improves the animals with the desirable qualities. That's because I can choose the animals and I can carry out the breeding techniques such that only the desirable qualities will be obtained and we get the progeny. So that's the reason why animal breeding is a very important aspect in animal husbandry, and we have done quite some research with with respect to these particular strategies on animal breeding. Now, before we try to understand how this is done, we should first know what exactly does the term breed mean. What does this mean? What does this mean? Now, breed is nothing but a group of animals. Breed is like a collection of animals, but the thing is, these are all related by descent and similar in most of the characters. Okay, like say their appearances or their features, their sizes, the configurations, their properties, all these are pretty much the same. Like in microorganisms, we use this term called as clone, where you will have a group of cells obtained from a single cell. Now, breed is very much similar to that. Here we will have one particular group of animals, all of them originated from similar type of animals. It can't be from single, it can be similar, and all this group of animals that's related by its descent, they have the same appearances, similar features, similar sizes, and also similar properties. So their yield or this disease resistance, all this are something which is very common. In fact, that's the reason why we have something called as breeds in cattle, many different types of breeds in poultry, and so on. Okay, now suppose there is breeding in between animals, uh, when we are talking about it, you see that breeding can be between two animals of the same breed, or it can be of different breeds. Now, if there is breeding between two animals of the same breed, we call them as inbreeding. What do we call them as? We call them as inbreeding. Now, what exactly is this? Now, basically, inbreeding is a mechanism, or it's, I would say, it as a technique where there is mating of very closely related individuals, which are, say, a part of the same breed. Okay, and this is done for around 4 to 6 generations. For around 4 to 6 generations, you see that there is mating between the same kind of organisms, and this is done when we want those characters to be maintained. Okay, now what we do here is, in the case of inbreeding, we are going to select certain animals for breeding. Now those animals which are selected for breeding purposes, we call them as super males and super females. Please do not confuse this with the super males and females you have studied in genetics. So these are the super males and the super females, and the super females of the same breed. uh, these are considered to be super males and super females mainly because their reproductive ability is higher. That is all it means. Okay, it is nothing to do with nothing to do with the breeding techniques or number of chromosomes and all that. They have a high reproductive ability. So you identify such animals, the super males and the super females, and they are mated in pairs. Okay. Now then what we would get is the progenies, and these progenies which are resulting from this cross, these are evaluated, they are checked, and then what we do is, in this progeny we are going to classify them basically into something like, the one which has a higher ability to reproduce, which we can always call them as the superior male, and superior males, and the superior females. So from super males to super females, many times we also call them as superior males and superior females. From there we are going to get the progeny. This progeny is evaluated to get or to select some of the most profound organisms which have higher reproductive ability, and we call them as the superior males and the superior females. Now these are allowed to mate further, and this gives the next progeny, the F2 generation, if I can call them with in terms of genetics. And then we do that for around 3 to 4 generations or 5 generations, and we are going to see that one particular trait which was there in the superior male and the female is passed on to the younger generation. Right. Now this kind of technique is what we call as inbreeding. Generally inbreeding is done in case of cows or buffaloes. That is mainly because we are going to select only those breeds, we cannot call strains, only those breeds which have the ability to produce more milk per lactation. Okay. We have one more male which is called as bull. Okay. Now bull is considered to give rise to superior progeny, superior males than those of other males. So like that, if you're talking about cows, we go for milk production, and if it is male, we go for sperm production, so on and so forth. And this technique is something that we call as inbreeding. Okay. Now if you remember what you have learned in your genetics chapter, you would have known that we have spoken about something that we called as uh, pure lines. We call them as the pure lines. Now what exactly are these pure lines? Pure lines are basically those you know progeny which do not have many different alleles at all in the first place. That homozygosity which we were discussing of. So all of them are homozygous for a particular trait, and this is something which is very very important. Why? Because even after 6 generations or 8 generations, you see that this homozygosity will ensure the passing of the genes, and you can see that the trait is observed in the generations. Right. So inbreeding is something that is very very important, because finally we want a pure line of genes in any animal. Now which pure line gives rise to what character? Now these are the things that has to be studied earlier. Okay. So what we do is we take certain characters, we have mapped the genes to them, and then from there uh, you see that we try to study about how the homozygosity condition would be, how the heterozygosity condition would be, and which condition would be the best, and then we are trying to separate them and try to see that the homozygosity is achieved, and finally we are having a homozygous group of animals. Now that is all possible only when we have inbreeding technique. So inbreeding is a very, very important and interesting method of breeding. Looks like, right? Because you can choose the characters and see that that is passed on throughout the generations. But along with that, you should know that it has some problems as well. Okay, now when you in the process of trying to see that there is homozygosity, in the process of trying to see that there is some kind of trait that is attained, the process of exposing the harmful recessive genes is high. Okay, right? So generally what you would see is whenever there is uh, some kind of natural breeding that occurs, there would be the presence of heterozygosity, or there would be natural selection, and therefore the recessive genes, the harmful recessive genes, are eliminated. But in the case of inbreeding technique, you see that those harmful recessive genes are harmful recessive genes are exposed. And what would be the effect of this gene exposure? Now that is something which for which you have to be ready with, because we are still not necessary, we still do not know how exactly would be the repercussions of this particular characteristic. So this is one bigger problem. Also, you should know that here in the case of in this particular inbreeding, we are trying to take only the superior genes. Yes, so I want gene for milk, or gene for production of more sperms, muscle mass, so on and so forth. And therefore what are we trying to do here is artificially, or with the involvement of man's efforts, there are certain genes that are increasing in number, and there is elimination of less desirable genes. Now if we keep on continuing this, okay, what would happen to natural selection? That is the first question. Okay, and since we are always involved into this, over a period of time, you see that since there is a selection at each step, which see the first progeny, try to take the best one, we go to the second progeny, there we to go to this identifying the best one, third progeny, identify the best one. So by the time we reach to around sixth generation, we have the best ones here. And this close breeding, or breeding within a very small population of animals, all of them which belong to the same breed. Please remember, when we're talking about the same breed, there will be only minute differences, and in those minute differences we are trying to pick and choose the best. Okay, now if we keep on doing this for some time, say around 4 to 5 generations, we do see that there is a reduction in the fertility. Okay, there is reduction in their fertility, and over a period of time you also see there is reduced productivity. Okay, so after around 6 to 10 generations, you do see that there is some kind of uh, fertility reduction, there is some kind of productivity reduction. Because of all this, you see that the traits might not be expressed, and this problem is what we call as inbreeding depression. Okay, it's called as inbreeding depression, and inbreeding depression is a very, very common sight in the case of plants as well as animals. And animals, we call it is more pronounced in case of cattle and poultries. Okay, so then what generally people do is uh, they take the superior males of some breed, and they will take the superior females of some another breed, and they try to breed, and this restores the fertility and the productivity. By chance there is inbreeding depression, what generally people do is they take the superior males from one breed, superior females from another breed, and these two are mated, and this next progeny would be a slightly different cross, mainly because it has a different breed altogether, and it has a cross, and then you see that the ill effects of inbreeding is overcome, right? So that is what we call as inbreeding. But again, when we are talking about the breeding, and when you have different good traits in different organisms, then we do not prefer inbreeding. We go for another one we call as which we call as outbreeding. You know what is outbreeding? Here we are taking unrelated animals. Okay, we are going to take unrelated animals, generally out of the breed. That is what it means. Not the same breed, but a different breed, slightly unrelated animals. um, or you see that we have sometimes breeding where we take two different species, or sometimes we go to a place where they belong to different families as well. uh, but this process of trying to bring about breeding between two different or unrelated animals is what we call as outbreeding. Okay. Just to explain types of outbreeding here, we have the first one which is called as outcrossing. It's called as outcrossing. Outcrossing, is nothing but the mating of animals within the same breed. Okay, it is from the same breed. But now, if it is in the same breed, it should be inbreeding. Here, it is not inbreeding, it is from the same breed. Then, how are they different? Here, it is something like uh, we do some kind of back study, and we get to know that from the past 4 to 6 generations or 8 generations, there are no common ancestors. Okay, something like uh, so around 6 to 8 generations back, there were two different breeds, and these two would have joined or mated together to give us one particular progeny, and this progeny would give rise to animals uh, showing characteristics of both the parents, and this has been happening for around 6 generations. Now if there is practice of having mating of animals within the same breed, but the thing is that these animals did not have any common ancestors on either side from past 4 to 6 generations, we call such a crossing as outcrossing. Okay. Now after we do the cross, we get the progeny, right? That progeny is called as outcross. We call them as outcross. Now we have lot of animals, which are all below average in milk production or beef production and so on. Okay, they're all below average. Now just because they're below average, we cannot kill those animals, right? And even if we kill, what to do with the carcass? So it has to be something like, it comes to use somewhere, and that is the time when we do outcross. We do one single outcross, and you see that it helps to overcome inbreeding depression. Okay, so outcrossing is something that is very commonly used to have to overcome inbreeding depression, right? Now, just like we have outcross, we have one more which is called as which is called as the cross breeding. Okay, so what exactly is crossbreeding? Now, in the case of crossbreeding, we are trying to take superior males of one superior males of breed A, let me say, is crossed with a superior female is crossed with superior females of breed B. So there are two different breeds, and we are trying to cross these two breeds. Okay. Now the progeny will uh, have the traits from breed A as well as breed B. Yes or no? Now this is generally used for commercial production. Okay. Uh, Sometimes you see that there is inbreeding and selection to new breeds and all that. Sometimes inbreeding is very difficult. Then we generally go for this kind of crossbreeding. Okay. Now this will be the progeny will be much superior to the existing breeds. Okay. There are many new animals or new breeds of animals that has come from uh, crossbreeding. Okay, especially in case of rams and ewes, sheep. Okay, we have many types of animals which have come into existence only because of crossbreeding. Now, just like we have crossbreeding, we have one more which is called as interspecific hybridization. Okay, we call this as interspecific hybridization. Now, what exactly is interspecific hybridization? Now, here we take a male and a female animal, and these male and female animals are from two different species. Okay, there are two different species which are getting mated. Now, if you know biology very well, you will get to know that generally a species are something group of animals which kind of link, which have the ability to mate together, right? Whereas if you have two different species, generally they will not mate at all. Okay, or even if they mate, their progeny would not be fertile. This is the whole idea of defining a species. But here we keep only the second one true and the first one is lost. So forcibly we see that two animals from two different species are mated, uh, and then the progeny would be would not be fertile obviously, but it will have some economic value. Maybe it has very high strength, physical strength, or it will have some or the other trait which has an economic value. And for this, the best example would be our mules. You know, mules. Uh, this is a cross between a dog and a horse, and has lot of physical strength. So people really use it to carry things from one place to the other. So that is what we call as interspecific hybridization. This is what we call as interspecific hybridization, right? So these are the different types of breeds and breeding techniques that are available right now. Now we spoke about how we can get different crosses, right? It can be in cross, out cross, interspecific, or intraspecific. We learned about all that. Now comes how exactly can we control this breeding, right? Now when we are trying to control the breeding experiments, you see there are so many types of methods that is available right now to us. We have lots of controlled breeding experiments. These are all carried out using one technique called as artificial insemination. Okay, so what exactly is this artificial insemination? So we have semination and insemination. What exactly is this? Now here we do collect the semen from from the male. Okay, now the male is chosen, uh, generally a superior male, but not necessarily all the time. We choose the best male. Okay, and then this semen is collected and it is injected into the reproductive tract of, again, the selected female. So we choose the male, we choose the female, take the semen and artificially inject it into the female. uh, so the semen is generally, you know, is we can use it at the same time immediately, but many times we do not do that. What we do is we collect the semen and we keep it in a frozen condition for a later date, okay, when it is to be required. So it is transported in the frozen form. So and where exactly there is a female, so there it is housed in the frozen form, because any animal sperm has a shorter lifespan, so it has to be frozen, and this way we carry out the desirable meetings. Okay, uh, so this is very much controlled breeding experiments, and the best part is that this also helps the breeder to overcome various problems of normal matings. So in the case of normal matings, there are various problems that would be encountered. Uh, so all these are kind of controlled by using this process of artificial insemination. Okay. Now you see that when we're talking about artificial insemination, we are taking the semen and we are injecting it into the female. This is what we generally do. Now, what do you think would be their success rate? Now, here the success rate is not very high at all, because the success rate is fairly low. You know why that happens? Because in the normal mating, animals are driven by natural instincts. Um, the length of the day, length of the night, the temperature, so on and so forth. Now, how much ever we have studied, we are still uh, yet to know a lot of other things that comes along with these animals, and therefore, you know, we have to find out that artificial insemination, though is a very nice uh, controlled controlled process of breeding, the success rate is quite low. In fact, that is the reason why we have CERT production of hybrids that helps to increase the success rate. Okay, we have one technology which is called as MOET. Okay, now what exactly is this MOET? It is called as multiple multiple ovulation embryo transfer technology. It's called as MOET. It is generally used for herd improvement. Okay, now you know what we do here is we take a cow. Generally the cow would be some cow which is fit to be a mother, has a higher reproductive ability. We select such a superior cow maybe, and then to this cow we are going to induce hormones. Okay, we are going to induce hormones. Now these hormones are like are having FSH like activity. What is FSH? We've already studied that it is a follicle stimulating hormone. So there would be release of eggs. So there is release of there is induction of follicular maturation. And because of all this, it's not just one egg that would be released, but there would be multiple eggs that would be released. So there is super ovulation. Okay, we call that as super ovulation. Therefore, just because of the induction of hormones like this, please do remember that this is also again by injection, right? Injection of artificially injecting the hormones into the cow. It does not release one egg, but it releases so many eggs, maybe around 5 to 8. Okay, uh, and because of this, you see a higher production or higher ovulation. And from here we do see that we do the artificial insemination, which is the animal is either mated with a bull, or generally we go for the artificial insemination method. And because of the either of these techniques, either the natural mating or by the artificial insemination, you do see that this particular process there is a higher chance of fertilization. Why? Because there is no one single egg, but since there is super ovulation, we have around 6 to 8 eggs, and at least 2 to 3 would get fertilized. So this is one particular process. Now then, after the fertilization happens inside the female reproductive system, the cell would form a zygote, and then the zygote would start dividing, and it starts dividing, and there would be a stage where there are around 8 to 32 cells. Okay, so there would be a stage where there are around 8 to 32 cells. Now these are collected these are collected again non-surgically, okay, and transferred to the surrogate mothers. Now the genetic mother is available for one more round of super ovulation. So this way the super ovulation process keeps on happening. There is development of all these fertilized eggs that is also happening, and you see that this technology helps to increase the number of cows or number of cattle. Okay, this is not only done only for cattle, it is done for sheep, it is done for rabbits, buffaloes, uh, mares, and many others. Also you see that very commonly we go for high milk yielding breeds of females, and lean meat with less lipid meat yielding bulls. All this are generally bred in a very short time and increase their herd size by this technique, which is called as MOET technology, or multiple ovulation embryo transfer technology. Now with all these techniques we do see that there is a sudden increase in the cattle, and also helps us to overcome the problems that is faced by the natural mating processes. So this way artificial insemination and MOET is kind of used quite often in case of all the animals like sheep, rabbits, cattle, so on and so forth. So this is all about breeding techniques. In our next class, we are supposed to learn about beekeeping and hive keeping. Okay, so bees and hives are also very important, uh, you know, as food. And along with that, fisheries is one particular area where there is a lot of improvement that still needs to be done. So that would be our topic for our next class. We will be talking about the beekeeping, hive keeping, and the animal fisheries. Um, and later we will jump to plant breeding technologies. So I hope you do not have any doubts. Any doubts, make a note. We will surely discuss about them in our live class. See you till then. Have a nice day. Bye.